Understanding force degradation testing in pharma. 6 months into stability, everything looked perfect until a new unknown peak appeared. No change in process, no change in packaging, yet the impurity kept growing. The question wasn't what went wrong. The real question was, why didn't we predict this earlier? That's exactly where the force degradation testing becomes critical. Hi, I'm Raghuveer Singh Pharma, Director Scientific Affairs at Resolve Mass Laboratories, Canada. At Resolve Mass, we partner with innovators to de-risk complex drug development through cutting-edge analytical strategies and deep expertise in challenging molecular modalities. Let's step into a common scenario. You have developed a promising molecule. Assay looks great. Impurities are under control. You move into stability, confident, but stability studies are slow. You wait months, sometimes years, to see how the drug behaves. Now imagine if you could accelerate time and understand how the molecule will degrade before stability even begins. That's the essence of force degradation. It's not just the stressing the drug, it's stimulating its entire life cycle under worst case conditions. You are essentially asking the molecule, where are you weak? How will you break? What impurities will you form? And most importantly, can my method detect them? What force degradation really means? In practice, we intentionally push the drug into uncomfortable territory. Acid, base, oxidation, heat, light, and humidity. Each one attacks a different part of the molecule. Esters often hydrolyze, phenolic groups oxidize, nitroaromatics can be light sensitive, amides may resist but degrade thermally. So force degradation isn't random stress, it's chemistry driven stress, and this is where many studies fall short. They apply standard conditions without thinking about the molecular structure. At Resolve Mass, we often start by reviewing functional group liability before designing the stress studies, because the molecule already tells you where to look if you listen carefully. Force degradation does three critical things. First, it helps build stability indicating methods. Your HPLC methods must separate API from degradants, not just known impurities. Second, it helps identify future impurities. The ones that appear after 12 months can often be predicted in weeks. Third, it supports formulation decisions. If oxidation dominates, the antioxidant hydrolysis dominates, control moisture. And the most misunderstood part, how much to degrade? Here is the question I often ask teams. Should we degrade as much as possible? The answer is no. The ideal range is 5 to 20% degradation. Less than 5%, you may not see meaningful impurities. More than 20% speaks starts overlapping. Beyond 50%, everything becomes messy. You lose resolution, peak purity becomes unreliable, mass balance collapses. We have seen cases where samples degraded 80% and the team concluded the method wasn't stability indicating. In reality, the study design was the problem, not the method. Practical study design. A good force degradation study is stepwise, not aggressive. Start mild, 0.1%, 0.1 normal acid, room temperature, short exposure, then increase gradually. This approach helps you control degradation and understand kinetics. Also, always test API, drug product, placebo, and blank. This is crucial because sometimes the impurity isn't from the API at all. We once evaluated the product where a major degradant was actually coming from excipient oxidation, not the drug. Without placebo stress testing, it would have been misidentified. Another critical step here is the neutralization. After acid or base stress, you must neutralize before injection. Otherwise, degradation continues in the autosampler vial. This is one of the most common real-world mistakes. Analytical evaluation. After stressing the samples, we evaluate peak purity, resolution, impurity profile, and mass balance. Mass balance is particularly important. You want to account for API loss plus degradants form is equal to 100%. If mass balance is poor, it tells you something is missing. Maybe volatile degradants, non-chromophoric impurities, detection wavelength mismatch, coelution. These insights help refine the method. At resolve mass, we often combine PDA and LC-MS during force degradation to confirm degradant identity and improve mass balance. This approach becomes extremely useful for complex or low-level degradants. Regulatory perspective. Regulators expect force degradation not as a checkbox but as a scientific justification. ICH-Q1A requires degradation pathway understanding. ICH-Q2 requires method specificity. Reviewers typically check where all stress conditions applied. Was degradation adequate? Is peak purity demonstrated? Is method stability indicating? Incomplete force degradation is one of the most common review deficiencies. And when that happens, approvals get delayed. Common mistakes seen in industry. Over-degradation. Single stress conditions only. No placebo testing. No neutralization. Poor documentation. Another subtle mistake, not monitoring time points. Instead of stressing for 24 hours directly, checking 1 hour, 3 hours, 6 hours help you understand degradation progression. This improves control and interpretation. So bringing it all together, force degradation is really about predicting the future of your drug. It helps you understand degradation pathways, develop stability indicating methods, support formulation decisions, and avoid stability surprises. Meet regulatory expectations as well. We are not just breaking up the drug, we are learning its behavior over time. The best force degradation studies don't just generate chromatograms, they tell the real life story of the molecule. And when done right, they prevent surprises, strengthen submissions, and build confidence in your method. If you are working on complex molecules or struggling with unclear degradation pathways, this is exactly where thoughtful force degradation design makes all the difference. Thanks for watching, and I'd love to hear what challenges you have faced in your force degradation studies. Thank you so much for watching this session, and see you in the next one. Goodbye for now.